All right, well, the ethical issue that we're going to be looking at today is that of biotechnology. Now, biotechnology is a really hot button issue right now. It's basically the field in philosophy where people still get paid to teach, right? Biotechnology, because of the advancements of medical science and in the area of cellular level research, genetics, all kinds of things like this. Because of those things, biotechnology is one of those fields where it is eminently relevant, where philosophy and ethics are eminently relevant. And so we're going to be looking at some various specific fields within the larger field of biotechnology, because really this subject or topic could cover anything from prosthetics to genetic engineering to harvesting and planting crops. Just all kinds of things can fall under this. But we're going to look at some of the more, I guess, hot button issues, if you will, some of the more controversial ones. But before we get started with all of that, let's go into some definitions on biotechnology. Some definitions. First of all, biotechnology in and of itself is the manipulation of biological systems using technology. The manipulation of biological systems using technology. Now, what do we mean by biological systems? Well, a biological system, quite simply, is any form of biological life. It can be something as small as a single-cell plant organism to something as complex as a multi-cell, multi-system organism. Anything along the spectrum. It could be an amoeba, it could be a human being, and anywhere in between. So the manipulation of a biological system using technology. So we use technology to manipulate those biological systems. We could do that on a genetic level. We could do that on a macro level, something like a prosthetic of some sort, and anything in between those two. Now, what is bioengineering? Bioengineering is the use of biological science to design machines and alter biological systems. So we will use bioengineering in order to advance the science of biotechnology. Now, there's another important distinction to make in this field of biotechnology, and that is the distinction between what we might call a therapy and an enhancement. A therapy and enhancement. Now, the difference is this. A therapy is when biotechnology is used in order to restore a biological system to its normal function. In other words, a particular biological system, it could be, let's say, a human being. A human being experiences some measure of disability. Maybe they've lost the use of their legs, for example. A therapy, when it comes to biotechnology, would be a way in which to restore as close as possible that biological system to its normal function. Now, in this case, a therapy may be something as simple as a wheelchair, something that allows the person to get around, even though they don't have use of their legs any longer, that might allow them to get around. Another form of therapy in this sense might be a form of stem cell therapy, which would allow that person to regrow the neurological damage that has been done to them, and then thus restoring the movement to their legs. It could be something on either end of the spectrum. But in any case, a therapy is that which is aimed at restoring a biological system to its normal function. Now, by contrast, an enhancement is something else that biotechnology endeavors to do. An enhancement is when biotechnology is used in order to push a biological system beyond its normal function. It's when biotechnology is used in order to push a biological system beyond its normal function. And this is for example, like performance-enhancing drugs might be a way of a kind of enhancement to a biological system. Now, between the two, between therapy and enhancement, typically speaking, therapies are not all that controversial, although there are some exceptions, and we'll look at a couple of those today. But for the most part, therapies are things that are not that controversial. These are things that are done in order to help others live a more fulfilled life, be able to be more self-sufficient, get around on their own, and do the kinds of things that anybody who without the disability might be able to do for themselves. So therapies usually are not all that controversial. On the other hand, enhancements are generally where we run into more of a controversial nature, things that are more controversial. This is, for example, we mentioned earlier performance enhancing drugs or therapies. They don't always have to be drugs. But for performance enhancing therapies, what would this do, for example, with athleticism? So back in the 80s and early 90s, there weren't as many regulations back then, and a lot of sports figures, a lot of people would engage in steroid use in order to enhance their performance. And they did. They got bigger, they got faster, they got stronger, they had more endurance. But as time went on, what we found is that these same kinds of therapies that were enhancements tended to have very negative side effects later on in life. They cause cancer and among all other maladies later on in life. And what made this particularly controversial is that whenever a person who did not engage in the use of performance-enhancing therapies or drugs, whenever such a person got on the field with people who were engaged in this kind of behavior, it was almost like there was no competition, and so it was really a pressure that the people felt, professional athletes felt at the time, that if I'm going to be out there doing this, and if I'm going to match the level of everybody else on the field, I'm going to have to be involved in these things as well. And so it led to a lot of people who normally would not have taken any of these things. It led to them actually doing it and harming themselves. And in the area of enhancements, there are also what might be known as cognitive enhancements. These are treatments or drugs that are designed to push the performance of the human mind past what they naturally would be capable of. And there's a real ethical question about whether we should do that or not as well. But so enhancements and therapies. So between the two, therapies and enhancements, typically enhancements are the ones that are more controversial. However, that's not always the case. And we're going to look at a couple of therapies which also have an additional layer of controversy to them as well. And the first one that we're going to be looking at is what it might be known as stem cell therapy or stem cell research. And there are some controversial ethical issues surrounding this as well. But in order to really get at what is the controversy around this, we have to understand what stem cells are and how they're utilized and how they're obtained in order to see the issue more clearly. Now, stem cells are what we might call undifferentiated somatic cells. Undifferentiated somatic cells. Now, the word undifferentiated basically means that these cells have no specific purpose at heart. They have no specific purpose. We have skin cells, we have heart cells, we have eye cells, brain cells, liver cells, kidney cells. Those have all been differentiated into the specific cell type to perform a specific function within the human body. But when we say that a cell is undifferentiated, what that means is that particular biological entity, that cell, has not been assigned and is not currently fulfilling a role in the body. It could become anything in the body. It could become a liver cell or a kidney cell or a lung cell or so forth. When we say it's a somatic cell, what that means, the word somatic means it is a cell of the body. Soma is a Greek word that means body or flesh. And to say that a cell is a somatic cell, it's a cell of the body. Now that is to be contrasted with what we might call a gamete cell. In a gamete cell are those cells that are used in sexual reproduction. A somatic cell has a full complement of human DNA in them. So within every somatic cell, a blueprint on how to build whatever creature that is included in every one of those somatic cells. However, in a gamete cell, you only have half the DNA that is necessary to form the animal. The other half would come from the other animal with which it's reproducing. So a somatic cell is a bodily cell that it contains a full complement of human DNA. So a stem cell is an undifferentiated somatic cell. Now, because it's undifferentiated, it can become any other kind of cell in the body when it reads that DNA, and then it can become whatever cell is needed. So that's what a stem cell is. Now, as far as where they come from, basically there are two broad sources, at least that is relevant to our discussion on the ethical nature of stem cell therapy. There are two sources of stem cells right now. On the one hand, there are what is known as embryonic stem cells. On the other hand, there is what is known as adult donor stem cells. Embryonic stem cells and adult donor stem cells. Embryonic stem cells are what we call pluripotent. And basically what pluripotent means is that they are so undifferentiated, they can become virtually anything else in the body that is necessary. Embryonic stem cells, as the name suggests, means that the source of these stem cells, these pluripotent stem cells, these are harvested from human embryos. Now, what is the source of the human embryos from which they are harvested? Well, typically speaking, embryonic stem cells are harvested from human embryos that are left over after in vitro fertilization therapies. These are fertility therapies where the mother donates a certain amount of eggs. She's given drugs so that she super ovulates and she can get some 10, 15 eggs. All of those eggs then are fertilized, and not all of those eggs will actually be used in the fertility therapy, and she may have some left over. She can then donate those to the use for stem cell research. So they are harvested from human embryos, and the source typically comes from in vitro fertilization. Now, I think perhaps we can see just for starters here why this practice can be controversial in the eyes of some. Because if you go back to our last lecture and remember that there are those who call themselves pro-life, and people who are pro-life have an aversion to bringing about the demise of any human being, no matter what stage of development they're in. Well, human embryos are human beings at a very early stage of development. Every single human being who is alive today was at one point in their life an embryo. An embryo, if you will recall, is a stage of human development that is after conception, after the sperm meets egg. The egg is fertilized. The egg moves down the fallopian tube and implants into the uterine wall of the mother, and then this primitive cell production begins to take place. Well, that primitive cell production is the production of stem cells. And the embryo produces all of these stem cells that are undifferentiated, so that as these cells produce more and produce more and produce more, they begin to differentiate out into and eventually form the human being, form the eyes and the nose and the heart and the lungs and so forth. They form these various systems within the human being. And so that is the embryo. That's the embryo. Well, these stem cells are harvested from those embryos, and that requires the destruction of these embryos. And so therefore, anyone who is pro-life would have to take pause and say, well, is this science a moral science? Because it leads to the destruction of human embryos, which in their view, in the view of the one who is pro-life, that constitutes the destruction of a human being. So those are embryonic stem cells. Now, the other source of stem cell therapy are what we might call adult donor stem cells. adult donor stem cells. And adult donor stem cells are what we call multipotent. Now, multipotent means that these stem cells are somewhat flexible. They're not nearly as flexible as embryonic stem cells, though. Embryonic stem cells can become anything you want them to, because they're pluripotent. Multipotent stem cells from adult donor stem cells, they can become a certain range of tissues, but not necessarily any tissue that you need them to be. So multipotent stem cells are flexible, but they're not as flexible as pluripotent. And these are called adult donor stem cells because they are typically harvested from sources of tissue donation. These could be umbilical cord blood. It could be bone marrow donation. It could be from peripheral blood elements, things that are extracted from the blood, etc. But there are all kinds of sources that do not require the destruction of a human embryo in order for adult donor stem cells to be utilized. Now, there are some other things about both embryonic stem cells and adult donor stem cells that we need to consider. Embryonic stem cells, as we've said before, are what we call pluripotent. And because they are pluripotent, they can become anything that you want them to. However, that same quality, the pluripotency of them, makes them somehow, some way, give rise to tumors in those who utilize those treatments. And because of this, there is currently no FDA-approved treatment utilizing embryonic stem cells out there. Now, there are all kinds of adult donor stem cells that can be used, all kinds of treatments utilizing adult donor stem cells. But currently, there is no FDA-approved treatment utilizing embryonic stem cells because they have been shown, because they're so pluripotent, they kind of get ahead of themselves and they can begin to cause tumors in the person who's been treated with them. Adult donor stem cells, on the other hand, because they are multipotent rather than pluripotent, they are less flexible. But that has turned out to have some great consequences, though, because of the lack of flexibility in them, they are more integral. They hold themselves better, and they do not give rise to tumors or cancer within the person who is utilizing it. Now there has been some recent developments, and by recent, I want to say this has actually probably been about 10, 12, 15 years ago. I'm not sure exactly on the date, but recent developments in adult donor stem cells, though, are this. Recently, science has been able to reprogram adult donor stem cells, which were multipotent, to become pluripotent. So they've been able to induce pluripotency from adult donor stem cells. And so it would seem that the necessity of utilizing embryonic stem cells is no longer a thing. Whereas before, scientists and researchers might have been somewhat beguiled or somewhat interested in using them because of their pluripotency. It is possible to induce pluripotency from adult donor stem cells. Once again, they can be made to be pluripotent, and as there are far more sources of adult donor stem cells than embryonic stem cells, it would seem that in order to do, if nothing more than just to get rid of the ethical dilemma that one faces. Utilizing embryonic stem cells, it would probably be a good idea just to get rid of that altogether and utilize only adult donor stem cells. But that is the issue of stem cell research. And a lot of really cutting edge therapies are being developed out of this, or being imagined anyway. They're saying that it might be possible to even heal someone who's been paralyzed. Quadriplegic. It may be possible one day, if the science continues at the rate it's going, that we might be able to allow do therapy to someone and allow them to walk again after they've not been able to walk for years, and so forth. Just all kinds of things like that as well. So anyway, some of the ethical concerns, obviously, with this whole topic are those that have to do with the sanctity of life. Harvesting some stem cells from embryos is often considered immoral because it requires the destruction of human life. Most embryos are donated from in vitro treatments, so yes, that is the case. They're not being utilized. However, that then maybe casts the question one step further and ask whether or not in vitro, because it creates more life than it actually utilizes, if that's a moral practice as well. Another moral issue to get into are the carcinogenic effects of the embryonic stem cells. If we give someone a therapy that we know is going to result in them getting cancer, why would we want to do that? So those are some ethical concerns related to embryonic stem cell research and stem cell therapies in general. The next issue that I'd like to get at is somewhat related, because sometimes people utilize this in order to develop stem cells, but that is the issue of cloning. Now, we typically think of cloning as something like, Attack of the Clones. It's Star Wars, it's science fiction, it's something like that. But in reality, cloning has already been accomplished on mammals, on higher mammals. Dolly the sheep being one of the cutting edge examples of this, and I think she was right around the late 90s, early 2000s. Scientists successfully cloned a sheep, and that was in the news for a long time. Sort of cutting edge pioneer science. But that's cloning. But cloning also is done for therapies or therapeutic reasons as well. And so that gives us two different types of cloning. Both of them utilize the science of cloning, which we'll talk about at a more basic level in a moment. But the two different forms of cloning, the first one is what we might call reproductive cloning. And this is cloning for the specific purpose of producing an entirely new being that is a genetic twin of whoever the cell donor is. So this is cloning for the purpose of producing a new being that is the genetic twin of the cell donor. This is what they did with Dolly the sheep. Dolly's cell donor was cloned from the person who donated, or the other sheep that donated that cell, and she was a genetic twin of that other sheep. Okay, so she was a separate entity, a separate being. She was a different sheep than that first one, but she was a genetic twin. Now, on the other hand, there's what we might call therapeutic cloning, and this is cloning for medical purposes. It is possible, in some cases, according to science, is desirable to obtain a clone in order to be used for medical purposes. So let's look at both of these, reproductive cloning and therapeutic cloning, in turn. First, we want to take a look at how does cloning work. How does cloning work? Well, this is for reproductive cloning. This essentially is also how therapeutic cloning works, except therapeutic cloning stops at an earlier stage. And we'll explain that in a moment. But this is how reproductive cloning works. In reproductive cloning, you have an ovum, which is basically an egg cell. And we're going to talk about this as if it were a human, okay? So this is an ovum, a human egg cell. And you can see in that ovum, you can see in the middle of that ovum that there's a small circle there. That is what we call the nucleus of the cell. And within that nucleus are exactly half the genetic material necessary in order to produce a human being. Now, if that egg cell is not fertilized by a sperm cell, that egg cell can never grow and develop and become a human. Now, once the sperm cell comes along, when the sperm cell fertilizes, the sperm, which is also a gamete cell, injects its half of the DNA necessary for the formation of a human being. The DNA from the sperm and the DNA from the ovum fuse together, and they become then they have a full set of human DNA within that nucleus. The egg cell reads that and then begins to grow and develop to build that human body. That's how normally reproduction works. Well, in cloning, what happens is instead of having a sperm cell to come and complete to fertilize the egg and complete the necessary genetic infusion that would give them a full complement of human DNA. What scientists do is they cut that nucleus out of the ovum. They remove the nucleus that is inside of the ovum. They take it out, and then they'll get what's known as a somatic cell. A somatic cell, which is a skin cell. In this case, this is a picture of a skin cell. Now that skin cell, you'll notice in the middle, also has a nucleus. The difference between the nucleus of the ovum and the nucleus of the skin cell is that the nucleus of the skin cell has a full complement of human DNA in it. It has all the necessary required DNA, whereas the ovum cell, the egg cell, does not. So scientists cut out the nucleus from the ovum, and then they remove the nucleus from the skin cell, and they put that into the ovum. Now, once that full complement of human DNA from the nucleus of the somatic cell is transplanted into the ovum, the ovum reacts as if it had a full complement of DNA, because now it does. And it reacts the same way that it would if it was fertilized by a sperm cell. And the egg then takes over, and the egg reads that DNA from the donated skin cell, and it creates a blastocyst. It starts building the human body. It creates a blastocyst, and then an embryo. It begins cell division. It begins stem cell creation and primitive tissue development. If it's allowed to continue, it continues on to become the fetus. And the fetus is basically a small human being in the embryo, in the amniotic sac. And then if the fetus is allowed to continue, the fetus eventually becomes a baby. And so that's how cloning works. So rather than the nucleus of the ovum being fertilized with a sperm cell, the nucleus of the skin cell is cut out of the skin cell, placed in the ovum, and then the ovum takes off from there and builds the body of the baby that eventually will be born. So that's how reproductive cloning works. Therapeutic cloning works in a very similar way. However, the process is interrupted before the fetus is formed. And so the same thing happens. The ovum, the nucleus is removed from the ovum. The nucleus from the skin cell is transplanted into the ovum. The ovum reads that full complement of human DNA, and it builds a blastocyst and builds an embryo. And then what happens is that it is not allowed to continue past that stage, because then the scientists, what they do then is they harvest the stem cells from the embryo that is being developed. And then those stem cells can then be programmed to develop things like pancreatic cells or nervous tissue or whatever kind of tissue that the scientist wants to have developed. That way they can direct the development of that tissue toward therapies that they can use. Now, in therapeutic cloning, one of the advantages that a person might have if this were perfected is that you could donate your own skin cell. And if you donated your own skin cell, then effectively the clone would be of you. You would be basically cloning yourself in order to use that tissue for whatever therapy you wanted to use it. Well, if you did that, the benefit of that is that there's really no chance of rejection. I mean, you're just going to put whatever tissue it is that you're going to be transplanting is going to be transplanted from your own DNA. It's going to have your own DNA, and your body will read that as if it were just any other DNA, any other part of your body, and it's not going to require the therapies that are necessary for anti-rejection and so forth. So that's how therapeutic cloning works. Now, what are some issues that are concerned with this, however? Now, obviously with therapeutic cloning, we have the same kind of issues that we have with stem cell research. With therapeutic cloning, we are destroying human embryos. And so anyone who is pro-life is going to, pretty much out of hand, is going to say, no, that's not a moral thing for us to do. But there are some ethical concerns, even if we were going to clone somebody in reproductive cloning, because there you're not actually destroying a human being, you're actually creating one. But there are still a couple of problems associated with that. Number one is the question of, are we not just playing God when it comes to this? Aren't we just playing God when it comes to wanting to create life? Aren't we tampering with the forces of this nature that we really should leave alone? That's a question that for some is utterly dismissed. But for others, really, it might give pause for us to kind of sit back and reflect on that for a moment. Really, is this therapy and advancing science in this direction, is that really worth it? But there are some more practical concerns rather than just sort of the philosophical concern, right? For example, one practical concern might be this. If we did clone somebody through reproductive cloning, and a person, we created a genetic twin of another human being, who does that clone belong to? I mean, we have several candidates here, right? For example, the person who donated the skin cell might say, well, that's my genetic twin, therefore the clone belongs to me, right? Whereas the person who donated the ovum, the egg cell, might say, well, you couldn't have done that. You couldn't have cloned anybody without my ovum. Therefore, the clone, the new clone, the new being belongs to me. But then the person, whoever it was that carried the child for however many, for 9 months and gave birth to the child, might say, yeah, but that child was inside me and my body was utilized to build that child. Therefore, the child belongs to me. But then it doesn't really even stop there, does it? Because then you might have somebody who comes in and says, well, the cloning process by which this child was cloned was patented in my name. Therefore, the child belongs to me. But then you might have financiers or people who back the whole project, people who gave grants or things like that, investors who might come along and say, no, the child, the results of this experiment were enabled by me. It was my money that made this possible. Therefore, the child belongs to me. And you can imagine if it were a corporation that did this, the child would be corporate property then, right? Or considered that way. So who does the clone belong to? There could potentially be a big uprising over that. And then last but certainly not least, there are the adverse or the potential adverse effects of the cloning on the individual who's cloned. Now, to understand this, we go back to Dolly the sheep. Dolly the sheep was cloned from a healthy sheep, and she was cloned and she was born and she lived for a while as a healthy sheep lives. However, what eventually brought about the demise of Dolly the sheep, and did so in a very rapid fashion, was that she experienced the onset of old age in a premature age. So she lived about half the lifespan of a normal sheep, and when she died of old age. So what happened? Why did this happen? What made this possible, and so forth? Well, here's the science of it. As we age, from the time that we are born until the time that we die, as we age, however long we age, our cells and our genes undergo a cellular aging at the epigenetic level. In other words, every so often our cells and our DNA is sort of recycled. This is a very layman's way of putting it, but our cells and our genetic information ages. And over time, it begins to break down. We begin to age at a cellular level. And so for example, at the time of this recording, I'm 47 years old. If I were to take and clone an individual from me right now, that child, when they were born, let's say they were born a year from now, that child would be born as an infant, but their cells would be 48 years old. And so within 40 years or so, they're going to develop all the kinds of maladies that are associated with old age, with a degenerating immune system, arthritis, all kinds of things that are associated with just the aging process. They're going to develop that at a much more rapid rate. because that cell, that DNA had been aged. And when it was inserted into that ovum, yes, the ovum was brand new, but we got rid of that genetic material, and the genetic material that we placed in there, the nucleus of my somatic cell, would have been 47 years old. So the child would not live to be very old, probably dying before they're 40 years old, 50 years old. In that of old age. So is it moral to bring a being into the world knowing that is going to happen, knowing that there's no way that they can escape that, knowing that we're going to introduce them into life rather than at the prime of life, they're going to have to go through life in age in a very rapid way. So there's possible adverse effects on the individual as well. So those are some ethical considerations with cloning. The next issue really, and this all sort of just kind of dovetails into one another, because stem cell research and stem cell therapies are made possible by cloning, and then cloning uses stem cell research as well. But then we also, at the genetic level, the next issue that I want to really just touch on briefly is that of what is known as genetically modified organisms, or GMOs. Now, typically when we talk about GMOs, genetically modified organisms, that brings up all kinds of images of the food that we eat, right? We usually hear that in association with plants that are genetically modified in order to increase harvest and make them more drought resistant and so forth. And that's really where it's used most widespread right now. There are places in order to make crops produce more, in order to make them drought resistant, in order to make them pest resistant, in order to make them disease resistant, they have been modified genetically. This is not only just the case with plants, however, that is also the case with animals. Now, in the past, both plants and animals have been hybridized. You get the plant or the animal with a trait that you like, and so you hybridize that with other plants or animals the traits of which you like, and you can create a bigger tomato or a meteor cow. But genetically modifying them takes them one step beyond a simple hybridization process, because a hybridization process can be utilized just by selective breeding. But when we're talking about genetically modifying, we're talking about cutting and pasting sequences of genetic codes that enable them to then go on and do things that they would have never been able to do had they not been genetically modified. Animals, for example, will produce more meat. Animals would be disease resistant, so you can keep them in close quarters and not have to worry about them developing diseases. They produce more young and produce at a younger age and produce it more often. Therefore, you can force the production of more meat that way. So food-based GMOs are really where the emphasis is nowadays. But that's not the only place where GMOs are right now being considered. Another sort of pioneering science right now are the development of what might be called chimeras. And a chimera is when genetic material from two or more organisms are combined to produce something entirely new. One specific example of this that is going on right now, that the science is developing right now, is in transplant therapies. Now, this is a therapeutic science, right? But it's a controversial therapeutic science. For right now, as of this recording, right now scientists are currently engaged in a project whereby they are splicing in human DNA into a pig's DNA, so that when the pig grows and gets older, it will develop a human heart. So they'll take the human DNA for the heart, and they'll splice it into a pig's DNA. Now, heart transplants, in the past, scientists have transplanted the heart of a pig and successfully put it in a human being for a little while. So the two are very close in size. They're very close in structure. And so now, instead of transplanting a pig's heart into a human, what they're doing is they're at the the germline level, they're splicing in human DNA into a pig so that when that pig grows and develops, it will develop a human heart in it, not a pig heart, a human heart. They're thinking, well, that human heart will be able to sustain a pig. Humans live longer than pigs and so forth. But what we'll do is we'll create a whole herd of pigs with human hearts, and then we can slaughter the pigs, take the hearts out, and use them to transplant into human beings. And that human being will have a human heart transplanted into them. And here's the deal. If we were to do this, it's possible. I mean, theoretically possible. I don't know that anybody's done this yet. But if the science is perfected, let's say I have a heart problem. Well, basically what I could do then is donate a somatic cell, just a swab off the inside of my cheek would do. The doctors would take my DNA sequence from my heart. They would cut that section out of the cell that I had donated. They would splice it into the pig cell, and they would create that pig specifically for me with my DNA heart already in that pig. Well, if that could be accomplished, then think about it. When that pig grows to adulthood, which is much sooner than a human would, then they could slaughter the pig, take the heart out of the pig, put the heart in me. It would have my DNA in it, so then it would not be rejected. So that's what might be known as chimera, and that's a science that actually is happening. And as of the recording of this video, I think scientists are up to the point where they have been able to achieve stage, and of course they abort these pigs before they're ever born. Just because they're in sort of a research phase right now, but they've done this, and the pigs are alive and still alive within their mothers, inside of their mother. Now, that's quite an interesting application of this science, but it does certainly bring up some significant ethical issues, both with the pigs and the chimeras, also with the issues of food GMOs. Again, there's the question, as there always seems to be in biotechnology, are we not playing God? And is it right? Is it moral to play God? Shouldn't it be such that we should not look into some areas of nature just out of respect for the supremacy of nature over us, rather than us trying to be masters over it? The second issue that we have, both on the food GMOs and on other forms of GMOs, chimeras, is that we really don't yet understand the long-term effects on either of these. And that's particularly concerning because a lot of GMOs are used in food that we currently consume. In fact, probably every bite of food that you consume today, unless you specifically went out of your way to select non-GMO items, probably everything that you ate today was GMO. It had GMO genetic modification done to it. They're already on the market. And a lot of people who object to GMOs say, look, we don't really understand the long-term effects of utilizing GMOs in food production and things like this. Another potential objection to GMOs are what we might call superbugs. Superbugs. And superbugs are these things, these bugs, these sicknesses or these illnesses that are resistant to whatever it is that we put on them. For example, let me just give an example. If we create a GMO plant that is resistant to normal diseases, then it stands to reason that those diseases will adapt to the new environment and become so strong that no sort of treatment that we give to them would knock them out. And those diseases then would go not only destroy our GMO crops, but because they're so strong, they would also then go and destroy our normal crops, our non-GMO crops. And that would potentially have a terrible environmental effect. And of course, you could say the same thing about pests and what kind of bugs eat these foods. You could say the same thing about it being drought resistant. There might be adverse effects to the plant, might be adverse effects to the nutritional value and so forth. But it's possible for these to lead to the creation of superbugs. And one last issue with GMOs that is different than the science itself, that is separate from the science itself, in terms of what effect it might have, is this, or what we might call patent issues. Patent issues. And now you know what a patent is, right? If you invent something, or if you have a process that you want to claim to, you go to the patent office, and it's basically a copyright for a process. And so let's say you invent a widget, and you go to the patent office and you get a patent for your widget, then nobody else can come and take your widget. It's considered your intellectual property, and you can do whatever you want to with it. You can sell it. Basically, you have the rights to that. Well, here are the patent issues. Imagine, if you will, on the one hand, on one side of the road, we've got a farmer who utilizes GMOs, and he buys his seed from whoever the producer of the GMOs, maybe it's Monsanto or who knows, whoever it might be, and he grows his field with GMO corn. Now, in the field next to him is somebody who is trying to do things the old-fashioned way, and so he specifically goes out of his way to buy non-GMOs. He doesn't want to spend the extra money. He'll go ahead and go through the extra processes that it takes to water it and keep the bugs out and keep the weeds out and everything. He doesn't want a GMO crop. Well, these two, they're on opposite sides of the road from one of these two fields, and when it comes time when pollination happens, well, the pollen doesn't discriminate between what field is GMO and what field is not. And so the pollen from the GMO field ends up in the non-GMO field, and the pollen from the non-GMO field ends up in the GMO field. Well, what's the problem here? Well, imagine you're the farmer with the non-GMO field, and you've got GMO pollen coming over into your field. Well, all that it takes is somebody from that company who has the patent on the GMO to go and buy a bushel of your corn and then have it tested, and it will be genetically linked to your GMO patent. Now what you can do is take that farmer to court because he utilizes, or his field has traces of the DNA of your GMO, patented GMO DNA in there. So companies have been doing this. They've been suing farmers for rights to their crop because their pollen somehow floated into the field of that farmer, and that farmer's crop has the DNA, the patented DNA in it. So that's another ethical issue, and that's more of a property rights issue, but one can see how messy this sort of thing can become. But those are some issues related to some moral issues related to GMOs. Well, don't forget to read your books. There are other issues certainly related to biotechnology that are of concern. Make sure to read your book. Check your syllabus for all the homework that's coming up. Make sure you get that done, and we will see you at the next video. All right, have a great day.